once again, welcome to Henry's Impact International Academy and at the time, and today we're going to be discussing viral replication. Alright, very good, let's dive right in. So, just as bacteria reproduce, virus also reproduce, but they do so making use of two major kinds of cycles. So, viral reproduction occurs via two major cycles. And what are the two cycles that we have? We have the lysogenic cycle and the lytic cycle. So, the lytic cycle, and then we also have the lysogenic cycle. So, majorly, the lytic cycle is just like an immediate infective cycle. So, it tends to just pull out its virulence. You understand? Within a shorter duration. So, it's just like onset of infection is short. So within a shorter period of time, the virus is already utilizing the host machinery system to rebuild itself, and then there is virulence, and instantly infection will start presenting itself. But in the lysogenic cycle, the onset of infection, which we call the OOI, is long. And that's why we say that this one experiences a large or longer period of dormancy. So we can just write it there, long dormancy. So there's a long period of dormancy. So it's dormant. You understand? At what point is it dormant? We're talking about as a result of the presence of what we call a prophage. So that prophage there is this phase here. I'll explain that to us just shortly. For clarity, let me just write it here. Dormancy. So this one's experiencing dormancy. Yes. So good. What we have here now are the step-by-step schematics of the viral replication. So this side here is where you have your lytic. This is the lytic cycle, which is the commonest cycle that you have. And then this is your lysogenic. The one that goes here and then has to come back to this is your lysogenic. Let me put it here. Lysogenic. This lysogenic from this stage can come back here, or from here can go into the infective stage. Can go or continue into the lytic stage, which is the infective. So good. What is step-by-step presentation of viral replication? So the first thing there you want to know is that there must be attachments, right? There must be attachments. So this viral phage, this is the phage. This guy here is called the phage. So this phage, in this case, a bacteriophage, because it's going to infect the bacteria. So yes, now, so you can see as it attaches itself, the next thing that's going to be happening there is that it's going to release this genetic material into the cytoplasm of this bacteria. And that's why you can see this green stuff here representing the nucleic acid, which is the genetic material of this bacteria found incorporated within its protein coat capsid. Yes, it gets released into the cytoplasm of the bacteria, and that's what you have here. So that means the stage 1, this is step 1, which is called attachment. So in the step 2 is the one that we call release. So that's the point where there is inoculation. We have the inoculation of viral genome into bacteria cell, or you can say into the bacterial cytoplasm, anyone. So that's what happened there. Now the next series of steps are very, very important. Now in the next step, this viral genome now will initiate the utilization of the host machinery in replicating. That is, it will enable it to actually initiate transcription and translation utilizing this host machinery. And that's why you can see that there is now aggregation of new protein coats here and the new attachment sheets, or what we call the hints. So here now, from here to here, you have that step 3. Are we there now? And this step 3 here, what you are really having in this point is transcription and translation. So it is utilizing, how does it go about this translation? It is utilizing what? What do we say? It will utilize the host's machinery to utilize the host machinery to form proteins suitable for integration, of new viral strains. So you can see that there. So that's what's really happening here. One other thing again that happens during this step 3 is that there will be hydrolysis. If you look at the plasmid of the bacterial cell, there is hydrolysis of that plasmid. You can see it's broken down into numerous fragments. So that's another thing that happens there. So during step 3, there is what? Hydrolysis. There is hydrolysis of bacteria plasmid. Yes, that happens too. So after you have that, there is now a presence of protein capsid and all these sheets, the neck and the hinge sheets. So what will now happen here is the next step, which is step 4. There will be assembly. So we have assembly. So assembly is where all the translated proteins now aggregate themselves, they come together, they integrate themselves to form these new strains. So that's what is really happening in assembly. It's an organized array. You're having an organized array of what? Of the viral components. So that's what you're having there. So after the assembly there will be large, I just drew 3 here. There will be numerous, millions of those viral strains. So what happens there is that after a period of time, it will initiate what we call lysis. So that's step 4. In that step 4, you're having what? You're having lysis, cell lysis. So cell lysis there is that point where the bacterial cell bursts open at a particular point, and that leads to the release of the new viral strain. So these new viral strains, look at some of them are having a copy of the initial bacterial plasmid and a copy of the inoculated viral genome, while some just have the initial viral genome. So the whole process can go and restart, where each of these guys can go and now attack a new bacterial cell. So this here describes the Lytic cycle. Let's come over to this place now. So right here, what you're having is the lysogenic cycle. So you just imagine now that the viral phage has been able to inoculate or release its genome into the bacterial cell. If that bacterial cell, rather than hydrolyzing if that viral genome, rather than hydrolyzing the bacterial plasmid, instead incorporates itself instantly into the bacterial genome, are you getting now? It will form what we call a prophage. That's when you know that this thing entered lysogenic. So the lysogenic is evident whenever there is formation of a prophage. What's a prophage? A genome or a genetic makeup formed by diffusion of both the viral genome and the bacterial plasmids. So that's what you are having here. So this guy here is called a prophage. So you're having a prophage here. How is it formed? Viral genome released and incorporates. So this one here, we're having the incorporation of what? Of viral genome into bacterial plasmid. Alright, so when that's done, it stays in this state for a long period of time, and then when it's ready, it stays in this phase for in some organisms it might be short period of time, sometimes long period of time. So after that period, it will start redividing itself. So that means it will be utilizing or allowing the bacterial cells to divide, so that when they divide, this same prophage, this new viral genome, will be present in both. Are you there now? So it continues division, continue. I guess now when we have large copies, they might start hydrolyzing, therefore releasing out their genomes. These the viral genomes that will now end up entering into this phase. You understand? It can enter. I told you that this lysogenic can come back into this place, or from here get into here. So that's it for your viral reproduction. It's that simple, not complex. Lysogenic and lytic. Make sure you do well to subscribe to our channel and also click on the notification bell, so when we drop more content like this, you will be notified.